Today's class we will discuss about the ATP synthetase enzyme and related theories according to this. So last time I told you about the diagram related to this. Now we had already discussed about ATP synthetase channel. Okay. Here electron carriers will be present, all that we have discussed. Let us not go back to that now. We know that. And here are the different types of rings. One is the C ring, correct? Above that is the A1 protein, correct? Then comes the stop also. Then you have alpha, beta, gamma. Sorry, gamma, epsilon, and delta. Like this. And then finally you have the alpha, beta sheets. Last class we discussed all these aspects. Alpha, beta will be present alternatively. We discussed all this. Remember all this, children? All of you remember this?
Yes, sir.
Okay.
Yes,
Where is the enzyme here? Enzyme will be at the base of the alpha beta sheets. Make a note of that. Here will be the enzyme. Enzyme is here. Okay. What is this fragment called as? Which is F naught in this? This or this? F naught and F1 fragments.
So F naught is on top, sir. F1 is below.
Okay, F naught and F1 fragments. We have discussed till here. I will not go back now. We have understood these aspects. Where are the protons present? Please make a note. This is alpha beta. Last class I had put in the message. Please draw the beta 2 protein. Beta 2 protein links the A1 series with the delta. Draw that one, all of you. All of you are done with this, understood these aspects. It's important for the examination. Can anyone tell me what topic this comes under?
Electron transport chain, sir.
After that, this is electron transport chain. This topic is different.
Oxidative phosphorylation.
Oxidative phosphorylation in exam. If oxidative phosphorylation is asked, highlight this. Last class I have given you some indications here. And mainly draw this ATP synthetase channel and explain about the concept of ATP synthesis. Okay, now coming back to this. So you have understood these aspects. What are the important proteins present and all? We will follow up this continuation now. See here, protons will be present here. Protons are present here. What is this? In a bacteria, what is this called? What is this space called? Between cell wall and cell membrane, this is. Protons will be present here.
So is it the periplasm?
Yeah, periplasmic space. If it is a eukaryotic mitochondria,
Intermembrane space.
intermembrane space. All of you have to be clear in this concept now. Intermembrane space. Okay. Irrespective of whichever it's prokaryote or eukaryote, protons will accumulate here and they will try to enter this channel. That's what we were discussing. So supporting to this, I have few things to tell you. So ATP synthetase enzyme we are talking about. It is present in this region. This is the enzyme. Yeah. Okay. To explain ATP synthesis, we have a theory called as Chemiosmotic theory. Note down this word. Chemiosmotic theory. Chemiosmotic theory was one of the important concepts given by Peter Mitchell. Peter Mitchell tells about this chemiosmotic theory. Okay, he is a biochemist. He is a biochemist. He explains about this concept of chemiosmotic theory. What's the meaning of this? He says that the protons, on the first point he tells us, protons in the space, it can be anything, the membrane space or periplasmic space. Okay, the protons in the space develops what? Low pH. Maybe low pH means it can be a pH 4, for example. So here it becomes low pH. Cytoplasm with high pH of 8, like that approximately. So down is pH 8. Here is pH 8. Make a note of this. Here is pH 8 and here is pH 4. This space is pH 4. All of you clear till here? pH 4, pH 8. So he calls this as pH variations. What is it? pH variation. Whenever such a pH variation happens, what the cell should always try to do? Related to this theory, it says something. It should show what? It should show what? It should show regulation. Should exhibit osmoregulation. What's the meaning of osmoregulation here? Protons to enter back into dash. Where should it enter? To regulate the pH, where should it enter? Any of you can answer? Try to
Into
guess.
the cytoplasm, sir.
Yeah, enter back to cytoplasm. So that the pH is regulated. The meaning is that. So, which is the channel it takes? It takes which channel? Dash channel. What channel does it take?
ATP synthetase channel.
Yes, I will call it as proton channel. You can always call it as proton channel or ATP synthetase channel. All are clear till here?
Yes, sir.
These points. Nityashri, Lavanya,
Yes, sir.
you understood this?
Yes, sir.
pH variation will be there. The pH has to be regulated. This concept all relatively is called as chemiosmotic theory. Who gave this? Who gave the chemiosmotic theory?
Peter.
Okay. So, proton channel is very important here. ATP synthesis channel is otherwise called proton channel.
Channel, sir.
Okay. Now, understand that. Come back to diagram. See the diagram, all of you now. When protons are entering the hollow core, it's a hollow core of proteins. When protons hit the A1 protein, they will bring changes in the protein, and of course they enter through the protein channel. When they enter the protein channel, they influence the A1 protein, C ring, beta 2 protein, stop, delta. Main connection is delta. Gamma, epsilon, alpha, beta sheets, everything one after another is influencing. Finally, the influence is what? What is this? Finally, the influence is
The
what?
enzyme, sir.
Enzyme also. So how do we put it across now, children? All of you follow till here, no? I'll drag the laptop. 1 second. So let me put it across here. Maybe I can use this board also. Okay. I'll use the side of the board. See here, I will mention as protons enter, protons will leave now. So the protons influence the whole channel. Protons will influence the whole channel. How to put across the points? You see the board now. First point. Protons enter ATP synthetase channel. For what? For what? Tell me. For dash regulation.
Osmoregulation, sir.
For osmoregulation. Okay. In the process, it influences on the proteins. In which order? A1, C, beta 2, gamma, delta, epsilon, alpha, beta. Everything will change, influence. This is called, can you guess? This is called, what is it? Read this.
Conformational changes.
Very important keyword. What is this? CONFIRMA.
Conformational changes.
Conformational changes. Okay, it brings a conformational change. That is more important. When it brings a conformational change, automatically it influences ATP synthetase enzyme. So first point is about all these things, chemiosmotic theory part. Second point is these things, about conformational changes. Thirdly, how the active site will change. Firstly, what you will talk about? Chemiosmotic theory. Secondly, what you will talk about? Conformational changes. You want me to send the pics of these? You will go through now. Shall I send you the pics of these?
Yes, sir. Yes, sir.
1 second. 1 second.
Yes.
Yeah.
Sir.
Huh? I'm sending. I'll send you in this one. WhatsApp only. I'm thinking I'll send everything together, one photo. 1 second. Whole thing will come. Once again, students, I'll send you. Make a note of the points, okay? Read it properly. Notes I'll send you. Notes means these only are the points anyway. I'm sending it. Can you see the WhatsApp? First is chemiosmotic theory, second part is conformational changes. Okay, no?
Yes, sir.
Okay. Now I will talk about the third part, that is about the enzymes active sites. Okay. Currently, what I am discussing is about the changes in the active site. Visible now? See here, enzyme is like this. Enzyme is like this. Got three active sites. It's got three active sites. Okay. So this is bound the ADP and inorganic phosphate. ADP and inorganic phosphate. How many sites are there? How many sites are there in the active site? In this enzyme, sorry?
3, sir.
3, sir.
Three active sites are there. Can it give more than 3 ATP? No. Isn't it? It can give only how many ATP it can give? The enzyme can give how many ATP to the maximum?
3, sir.
ADP plus ATP. See, it is not binding at all. It has not formed ATP. It's not formed ATP yet. This state is called O state. Sorry, low state. Low state. L state means loose state. Loose state. Okay, this is called L state or loose state. This will not form ATP yet, it will be there in the same. Understood? After protons enter, see the aroma. Protons enter, what is the change it will bring in the protein? Huh? After conformational change, only then the active site will change. Understood? So now we should draw the diagram. Diagram changes. See? It's called tight state. Tight means the modification of the enzyme will be there. Just won't change it. See? This is called tight state. Already ATP is formed now. See? This is ATP. This is ATP. Like that, ATP is formed. Like this. Got it, all of you? Could you differentiate both the diagrams? See both the diagrams. Only after conformational changes, ATP active site changed. Active site of the ATP synthetase changed. Understood? What is the state called as? T means, what is it? What state?
Tight state, sir.
Tight state. Yes, good. Once they are bound to become ATP, it has to release itself from there. So what will happen? See the element again. I am drawing it this side. Drawing it above. I have drawn this. Did you understand the concept? So
Yes, sir.
as protons leave the channel, completes osmoregulation. So osmoregulation is over. Then again enzyme will change. See, enzyme is back position. It's back to position. Again, it's become bigger active sites. This is called as O state. Sorry, O means open state. O means open state. What is the meaning of open state? As protons leave the channel, ATP is released freely. ATP is released freely. New ADP will come and bind. These are new ADPs. New ADP and inorganic phosphate binds. Same way here also, in all the places, I just shown one. In all the places, what we should show? ATP is free. Huh? ATP is free. Like this, you should understand. So, 3 ATPs are forming in different active sites. All are clear till this?
Yes, sir.
Yes, sir.
How many states are there? L state, T state, O state. What is L state? Tell me, someone tell me what is L state?
Loose state, sir.
Oh, what happens here? What
Here,
happens there?
so the ADP and inorganic phosphate have not bound together, sir.
They're just present. Okay. After conformational change, what happened? What difference? ADP became
ATP, sir.
ATP. That is called T state or tight state. ATP is free. How is it free? Once the protons leave the channel, phosphorylation is over, active sites are back to normal. That time ATP is free. And what are these? What are these?
So new ADP and inorganic phosphate, sir.
New ADP and inorganic phosphates have bound. This is called as O state or open state.
Open state.
This much you have to understand and write in the exam for oxidative phosphorylation. Last class we discussed about the diagram and the diagram. Okay, this diagram.
Sir,
Yes, ma'am.
excuse me, sir. Sir, in open state, the ADP doesn't bind. In open state, there is only release of ATP molecules, right? Free ATP.
In open state, it's a transition stage, ma. ATP is released free and new ones will bind. That means it's again coming back to L state. It's understood.
Okay, sir.
O becomes L again. Got it? It's a transition state. Open state will be release of ATP. Once ADP binds, it again comes back to what state? First one. L state. LTO keeps on repeating. Clear?
Yes, sir.
When will T state come? Only when proton motive force is there. Please mention that also here if you want. Conformational changes are written now. They are written in PMF. What is PMF? Without proton motive force?
Proton motive force.
Yes, no conformational changes happen. That also you can mention here. Shall I send a pic of this part? You will all draw it.
Yes, sir.
Yes, sir. Yeah. If I missed out something, please draw that properly. One second, students, I'm sending it. Please check. I've sent you. Okay, no?
Yes, sir.
Please tally everything, understand it. It's an important part of the examination. Go through properly. Oxidative phosphorylation will be thorough. Even electron transport chain, part of it, some questions will come. Be thorough in the electron carriers also. Okay, students. Connected to this topic, I have oxidation of inorganic compounds. I'll be talking about that topic now. I'll start with that topic. Shall I clean the board? Everything is fine with this till now?
Yes, sir.
Okay, I'll continue with the next topic. Give me 2 minutes, I'll clean the board. I'll push this side one second. Okay, so I'm coming back to this side of the board. I'll be talking about oxidation of inorganic compounds. Write the heading. Oxidation of inorganic compounds. See, what's the meaning of this? Inorganic compounds mean we discuss about ATP generated by using inorganic compounds. So certain organisms produce energy by using inorganic compounds. Okay, these are called as chemoautotrophs. They are called as chemoautotrophs. This is also called chemoautotrophy. Chemoautotrophy. Many question papers you have found this word chemoautotrophy. Chemoautotrophs. What they use only inorganic compounds. I am discussing few things according to your syllabus. Like say for example, some of them will use nitrogen. Some of them will use nitrogen. Okay, in nitrogen you have two things. Nitrosomonas and nitrobacter. Okay, and some of them will use iron. Iron. Okay, I'll come back to each one. They use iron example Thiobacillus. Are you writing? Yes. Thiobacillus peroxidans. Then some of them use sulfur. Sulfur oxidation we call it as. Example, Thiobacillus, same genus, Thiobacillus thiooxidans. Sulfur. Thiobacillus thiooxidans. Same genus. Even sulfolobus. There are certain organisms which use hydrogen gas. Example, hydrogenomonas. Hydrogenomonas. These are the different organisms which use different types of inorganic compounds as source of energy. Nitrogen, iron, sulfur, hydrogen. These four are there in your syllabus, children. You have to study all these four in your syllabus which is there. Okay. What is this called as? Chemoautotrophy. One of the topics I will discuss with you for time being. Let me take up. There is a few minutes left. I think I will go with iron only. Okay. Nitrogen will take time. I think I have 8 minutes or something. I will discuss that one. All are understanding this? Yes.
Yes, sir.
Yes, sir.
This can be a 2 marks. Initial question can be 2 marks. What is team order 2, 3? Each question, whatever is there, each topic, whatever is here, can come for either 5 or 2 or 1. We don't know how they will give. And then you practice the diagrams of all these 4. All like ETC diagrams only. Electron transport chain diagrams, what you studied no earlier for ETC chain, like that it will come. So let me draw one of them now. Let me take up iron oxidation, just simple because we can complete it for today. Next class we will now go with nitrogen and sulfur and all. Shall I clean the board? Yes.
Yes, sir.
Okay, let us say for example, I am taking up iron oxidation as one of the topics. Iron oxidation, it means oxidation of iron. Okay, remember in this topic, I'm talking about thiobacillus peroxidans. This bacteria is usually found as one of the organisms in the acid waters, acid mine waters. Okay, so I'm talking about mining process, application of technique, introduction of technique. I'm talking about mining process. When they do mining, there are some acid waters coming up. Okay, these acid waters, whatever I'm discussing, these acid waters will have the bacteria. Like what? Thiobacillus, peroxidans. This bacteria will be present in the acid waters. The pH is very less that time. pH is around even 3. In such an environment, you can find this organism. This organism naturally also can remove the metals. So for example, I'll give you an example here. CuFeS2 is one of the ore particles. It's a low grade ore. Can anyone tell me what's the meaning of a low grade ore? What's a low grade ore? What is an ore first of all? Ore is a soil which has got a desirable metal. Correct, no? Ore is something which is of a desirable metal. Got it? So, copper is required now. Copper we have to recover from this ore. Okay, worldwide they're using this technology. They're using. Have you heard of this topic? What is this? Huh? What is this?
Leaching.
Leaching. Bioleaching means what? What's the meaning of bioleaching? Huh? Write down. Recovery of metals from ores by using microorganisms. By using microorganisms. That is called bioleaching. Why am I talking about all these things? This bacteria, Thiobacillus peroxidans, uses iron. When they use iron, they release copper free. How to understand this? Did you take down these facts, all of you? Do you understand these parts? Imagine there are rock particles here. Ore particles, I mean to say. Broken ore particles. It's a broken ore. Okay, in this broken ore, what they do? They sprinkle water. They spray water. Okay, and I'll give you the procedure. Acid water, these are waters which will come up. Later on becomes acidic water. I'll tell you how. It will time later. So there's acid waters here. Copper gets deposited here. Copper gets deposited here. And this acid waters can be again pumped back. These acid waters can be again pumped back. so that bioleaching will continue. So bioleaching process. These are broken ore particles what is taken. This is an application. You will be studying this again in your final year. This part, what is bioleaching or not, you will study again. But try to understand why I am talking about this. Whenever a bacteria use iron, they copper or gold or whichever metal is there, it becomes free and it gets settled down in this. This is called launder. This instrument is called launder. This is called a bioreactor. So first they break the ore particles, put it into the bioreactor, sprinkle water, the water will percolate. In the process, some chemical reactions happen. I'll discuss that points now. Copper gets deposited and we can use acid waters once again to make the process more faster. Did you get the basics, children? All of you got the basics of this? Huh?
Yes, sir.
Okay. Now I'll give
Yes,
you one
sir.
reaction. All of you copy down the reaction. I'll write it on the board here. This is followed, no doubts in this, no? All of you got these points. Yes, Dviti. Dviti, you followed?
Yes, sir. Yes, sir.
Followed everything. Did you follow this concept? Right on.
Yes, sir, I followed.
I'll send you some pics after this. We'll just go through. FeS2 plus water. Right on. FeS2 plus water plus sulfur dioxide. Plus sulfur dioxide gas. Gives FeSO4 plus sulfuric acid. FeSO4 plus H2SO4. FeSO4 plus H2SO4. It gives that. See, got acid here. Imagine copper is here, okay? I'm giving general reaction. Copper and copper you can show here if you want. So this is a ferrous sulfide, ferric sulfide converting to ferric sulfate. Sulfide giving sulfate. In the next reaction, please check the attendance, ma. Meenakshi. I don't know how much time is there. Hey, less than 1 minute, Baba. Please check the attendance.
Sir, I took it, sir. I
Okay.
took it, sir.
I'll send you this reaction, children. Go through that different the class dealings. 2 FeSO4 means ferrous sulfate, ferric sulfate plus sulfur dioxide.